The overall goal of this procedure is to create transient pores in cell membranes using inkjet printing. This is accomplished by first modifying a standard inkjet printer. The second step is to empty and clean a stock printer ink cartridge to allow for the insertion of the bioink. Next, the cells of interest are suspended in a solution of fluorescently tagged actin monomers and PBS to create the bioink solution. The final step is to print the bioink onto microscope slides using the modified printer and cartridge. Ultimately, fluorescent microscopy can be used to show that the fluorophore conjugated G-actin monomers have been internalized by the printed cells through transient membrane pores.
The main advantage of this technique over standard transfection and cell microinjection techniques is that this printing technique is easy to use and yields high cell viability.
Though this technique can provide insight into genetics, it can also be applied to other fields of study such as cell biophysics and tissue engineering. In cell biophysics, this technique can be used to easily watch the cytoskeleton of a cell during cell migration.
We first had the idea for this method when we wanted to be able to visualize changes in cytoskeleton arrangement due to applied forces on cells.
Begin by unlocking the plastic clips from the bottom base of the printer and then slowly lift off the top plastic case. Then detach the button display light panel from the top of the printer, leaving it connected to the printer's motherboard. Using Kim wipes and ethanol, clean the inside of the printer, especially the areas where the ink cartridge rests and where printing occurs. Next, locate the cables supplying power to the paper feed mechanisms. For the HP DeskJet 500, they are just below the paper tray to the left of the front, and unplug the cables from the motherboard. Now locate the paper detection mechanism.